I scored a 524 on the Mcat, and here is a quick, high-yield summary of the electron transport chain and oxidative phosphorylation. These pathways occur within the inner mitochondrial membrane in eukaryotes, and in prokaryotes they occur within the cell membrane. The electron transport chain has four major enzyme complexes: complex 1, 2, 3, and 4. And complex 1 receives electrons via NADH, whereas complex 2 receives electrons from FADH2. And these are electron carrier equivalents that are produced in the course of other metabolic pathways, such as glycolysis or the TCA cycle. Ultimately, these electrons are shunted from complex 1 and 2 to complex 3, and subsequently from complex 3 to complex 4 via a series of oxidation reduction reactions. And complex 4 will shunt these electrons to oxygen, which is the final electron acceptor. Overall, this process of oxidation reduction reactions is exergonic, so the delta G is negative. It occurs spontaneously. And this releases energy. That energy is harnessed by complexes 1, 3, and 4 to pump protons against their electrochemical gradient from the matrix of the mitochondria into the intermembrane space in eukaryotes. The proton gradient that is established in this way is subsequently used for oxidative phosphorylation, which is mediated by complex 5, aka ATP synthase. Now, ATP synthase is a complex structure which contains essentially a channel that allows protons to run down their electrochemical gradient from the intermembrane space into the matrix. And it subsequently harnesses the kinetic energy of proton motion in order to catalyze the reaction of ADP plus inorganic phosphate into ATP. And that movement of protons down their electrochemical gradient is referred to as the proton motive force. Interestingly, uncoupling agents such as 2,4-dinitrophenol are drugs that can bind to protons within the intermembrane space and shunt them across the inner mitochondrial membrane independent of ATP synthase, thus dissipating the proton gradient and reducing the proton motive force. Back in the day, such drugs would be used as weight loss agents because they would reduce the amount of ATP synthesized per glucose molecule and, more importantly, per lipid molecule, thus burning fat. Check out our Discord community for more.